Thank you. The first thing we want to do today is draw the structures of DNA and RNA. Are you ready? So we're going to do DNA first. Which one has what? The 3-carbon. Oh, it's a 5-carbon sugar. 1, 2, 3, 4, and the 5th one's up here. Now this is getting into some real chemistry. I'm going to explain what this all means. But because this is DNA, the DNA obviously stands for deoxy. Yes, that would be great. Are you drawing in pen? Yeah. That's risky.
Are you gonna read?
I might. I'm trying to remember if. Hold on.
Wow.
No, it's right. That's right. And yes, I'm going to explain what this all is. But there's also, we know that this is, I guess I should do. There she is. Hi, Selena.
Hey, good morning.
Good morning. We're just drawing the structures of DNA and RNA right now.
Okay.
Now, here's a tip. In order to see better, like to see what I've drawn on the board a little bit better, you can click on the pen, like pen to screen button, and that'll make it bigger on your screen. Do you see that, Lexi?
Mhm.
Okay, cool. And is it backwards? No. Okay. Okay. So they're both 5-carbon sugars. They're both pentoses. You can see that they are sugars because they end in OSE. And then pent. Pent is a prefix meaning 5. And we can see that they each have 5 carbons. 1, 2, 3, 4, 5. And this is how we get the term 3' to 5' direction, or 5' to 3' direction. But notice that the oxygen always points towards that 5' direction. Cool? Okay. Now, do you remember there's a term called dehydration synthesis? Do you know what that's called, or do you know what that is? Do you remember?
Oh, when they take the water out, you make
Excellent, yeah.
that was this class, right? Not chemistry.
Yeah, that was way back in like September. So that means to remove an OH group to make a polymer, or to remove an OH group to make a polymer. So in this case, we can go in and we can remove this OH group to add the phosphate group. Or we can go in and remove this to add the nitrogen base. A, T, C, or G. And over here, we can remove this one to add A, U, C, or G. Got it? Any questions about it? Okay. So the opposite of dehydration synthesis, do you remember what that's called? Good. Or well, hydrolysis. I guess you could say hydration synthesis. I don't know that. Well, it wouldn't be synthesis because synthesis means to make something. The opposite would be, of course, to break something apart. So let's say we added this phosphate group. If we then wanted to go back and remove it, we would need the process of hydrolysis. But it's just important to note that the difference between deoxyribose sugar is right here. If you notice, we removed an oxygen. Can you see that? Yeah. Okay. And so really they're really two different things. This is deoxyribose sugar. This is ribose sugar. Okay. Next slide.
Can you scoot over so I can write my bad?
This is unit 6. Yes. Selena, let me know when you're done drawing.
All right, I'm finished.
Okay, so now we want to focus on the different types of RNA, which you can see on the screen. We don't talk much about ribosomal RNA in the standard biology class, but we do talk about messenger RNA, and we do talk about a little bit about transfer RNA. Somebody took my, I wonder where those are. Okay, so let's talk about each of them now. They involve the same nucleotides, though. They all use the same nucleotide with the ribose sugar and with A, U, C, and G bases. Here's an image that kind of shows the difference between the three and how they're used in the process of transcription and translation, the process of protein synthesis in general. We'll talk more about it. Messenger RNA is the first one that comes up in this process. It carries the message from DNA to the ribosome. We'll talk more about that. Transfer RNA comes second, and transfer RNA's job is to bring amino acids to the ribosome. And then ribosomal RNA is last, and it's used essentially just to make a protein. Okay. So mRNA stands for messenger RNA. mRNA carries a message, a genetic information message, from DNA to the ribosome. Of course, in a eukaryotic cell, the DNA would be located where? Where do we find DNA in a eukaryotic cell? Yes, good, Lexi. We find it in the nucleus. mRNA has a linear shape, which means that it's in a line, essentially. It consists of codons, which we can kind of see in that image on the right. And a codon is just a group of 3 nucleotides. And mRNA can range in size pretty widely, from anywhere between 400 and 12,000 nucleotides in length. So am I good to erase this? I'm just going to erase the deoxyribose.
Yeah.
Okay. Lexi, can I erase this? So can one of you read me the sequence here?
From the top to bottom.
Yeah.
G, A, C, U, A.
Wait, G, A, C, U,
A,
A,
C,
C,
G, A, B, C, E, U, P, G.
okay. Where are we?
Put on side of the first G.
C, G, G. All right, I'm just going to stop there because I'm out of space. All right, so we have 5 codons here. Codon 1, 2, 3, 4. Uh-oh. G. Wait a minute. GCU. Let's see.
Yeah, G, C, U, U, C.
GCU A.
Yeah.
How did I end up with this?
I don't know. I don't want to see your hard hearing.
Okay, let's try this again. GCU.
A, C, G,
Yeah.
D, A, G, P, U, U, P, G, G.
Okay.
A.
Thank you. So we're just going to do 5 codons here. I'm going to roll this over. So we've got 5 mRNA codons. Now, how did we get these mRNA? Can you tell me the DNA that would have given us this mRNA?
C,
C,
P, A, P,
good,
G,
good,
C, C, P, C, G, A, G, G.
good. And so going from this to this is called what? What's that process called? Do you remember, Selena? What's the process of going from the DNA to mRNA?
Um, is it transcription?
It is transcription. Good. We're going to talk about it a little bit later. Transcription. I can always remember this, or I can remember the order because transcription is alphabetically before translation. So transcription comes first. Okay. Were you able to write down all these bullet points? Okay. Selena, are you good to go here?
I need to write mRNA.
Okay.
Well, I just took a picture of it, so if you want to erase it, you can.
No, I'm just talking about, did you write down what's on the actual slide, not what's on the board?
Oh, yeah, I did.
Okay, cool. Okay, then there's transfer RNA. Transfer RNA has a clover or a loop shape. It kind of looks like a lowercase t in my mind. We're not going to actually talk about the different segments of this tRNA. There are different names. There's a T arm and a D arm, and these things we don't really. It's above the scope of this class. But the role of this transfer RNA is to bring a specific amino acid from the cytoplasm into the ribosome. This is a pretty cool process. So the tRNA has what's called an anticodon at the bottom of it. That anticodon is going to correspond directly to the codon of an mRNA transcript. I'll explain exactly what I mean by that shortly. But it matches up with a specific codon on mRNA. And each specific transfer RNA, based on its anticodon, is going to bring in a specific amino acid. Okay, so let's take a look at this. Let's just do the first. All right. So we've got a transcript of mRNA here. And it is G, C, U, A, C, G. I should have moved that over. G, C, U, A, C, G. Okay, so we've got an mRNA transcript. I'm just going to do the first two codons. Now we want to think about what might be on the tRNA anticodons. So the anticodon is basically just the complementary codon, or the complement of the codon. So what I mean by that is, what letter of RNA corresponds to a G, or is it's going to be a C. Then what?
G, A,
Good.
U,
Good.
T,
Good. So here are your two anticodons. And each of them is going to bring with them or bring with it a specific amino acid. Here's your tRNA. Here's another one, tRNA. And up here, there's going to be an amino acid. I'll use a different color. So it's the anticodon that allows the tRNA to know which amino acid to bring. And they're going to bring these amino acids from somewhere in the cytoplasm. Keep in mind that the cell is always taking in new things. So your blood cells might take in some type of virus and break it down. And so the proteins that made up that virus, once they get broken down, can then be repurposed, right? We can break it down, and if it was made up of 5 amino acids, and that's really it would be like 5,000, but let's just say it's made up of 5. Once we break it down, we can use those same 5 amino acids to make proteins that the cell actually needs. That's why there are always amino acids kind of just floating around in the cytoplasm. Does this make sense?
Yeah.
Selena, do you have questions about that?
No, I was just listening.
Okay. And then there's one more type of RNA. There's ribosomal RNA. I'm not going to spend too much time. This could be a really complex conversation, so I won't spend too much time explaining this. But ribosomal RNA composes the ribosome. So basically the ribosome is made up of RNA. There are some other proteins that help to make the ribosome, but it is largely made up of RNA. It has a spherical shape, but it has 2 sections. There's what's called a large subunit, which kind of goes on top of that mRNA transcript. And then it has a small subunit, which is below that mRNA transcript. And the way that the shape or the size can be explained is also complicated, so I won't get into that right now either. This is so much easier when I can draw. You got it, Lexi?
Yeah.
Okay, Selena, let us know when you're ready to go.
Yeah, I need to write what's on the board.
Okay.
Okay, I'm finished.
All right, so the central dogma of biology is this: DNA nucleotides get transcribed into mRNA. And later we'll talk about the process of translation, in which that mRNA transcript is used to make a protein or to make the primary structure of a protein. Fancy colors.
I'm finished.
Cool. So I've always thought about this process of transcription as being similar to hearing a language spoken and writing that same language on paper. So instead of just kind of reproducing it in the same medium, so instead of just like recording the voice, you're actually going to write it down. So you're transcribing it. It's not coming out in the same way. It came out initially from a voice, and then you actually put it on paper, carrying the same message, and it's written in the same language. This is similar to this process of transcription. We're writing DNA and RNA, they're written in the same language, in the language of nucleotides. A's and T's and C's and G's and U's. It's the same language, but it's slightly different. Slightly different. Okay. So here's the process of transcription. It's not really all that complicated. In my mind, it's not nearly as complicated as DNA replication, which we talked about last week. So I won't ask you all to draw this picture. What I do want is for you to write down these blurbs, or at least, paraphrase. So for each gene, only one of the two DNA strands is transcribed. And of course, we've got to remember that a gene is just a small section of DNA. The strand that ends up being transcribed is called the template strand. Could also be called the non-coding strand or the nonsense strand, but we're going to call it the template strand. You good, Lexi?
I'm finished writing.
Okay. That's fast. Okay. So it could either one of the strands of DNA. Of course, obviously DNA is a double helix, so it has two strands. Either one of them can be transcribed. It's really just a matter of which gene is being transcribed at the time. So whichever one gets transcribed, we're going to read it in the 3' to 5' direction. And I'm going to explain why that is. But the thing that is actually doing the reading is that big purple enzyme in the background called RNA polymerase. The strand that does not end up getting read, or the strand that is not transcribed, is called the coding strand. Okay, so let's say we've got a strand of DNA that looks like this, right? It's obviously a molecule of DNA. I keep saying that, but it's got two strands, obviously. One is going, they're anti-parallel. So the 5' of one is next to the 3' of the other. We've gone over this. Which one do you think we should transcribe? The top or the bottom? Okay, so you want to transcribe the bottom. It's always going to be transcribed in what direction? What direction does it get read? 3' to 5'. Yes. So that means that RNA polymerase is going to read it this way. And of course, we see this ASE ending because it's an enzyme. You can. I just want to demonstrate why this is happening. So this strand now becomes the template strand. And then what's this one called? The coding strand. You might be wondering, why is this called the coding strand? And we are about to see why. So RNA polymerase is reading it, and it's adding new RNA nucleotides in this direction. So if this is the 3' direction of the DNA strand, then which end of the RNA strand is this going to be? They've got to be, yes, they've got to be anti-parallel. So this has to be the 5' end. Now it makes sense as to why it has to be read 3' to 5'. Remember, we can only add new nucleotides to the 3' end. Does this make sense? Okay, good. Thank you. So we've got to transcribe in the 3' to 5' direction. We've got to read the DNA strand in the 3' to 5' direction. The reason for that is because the new RNA is going to be anti-parallel to this DNA. So when we start off on the 3' end, we're making new nucleotides from the 5', but we can only add on the 3' end of each nucleotide. So we've got to keep going this way. I'm going to add a nucleotide here, which is going to be A. So its 3' end is over here. I can only add to the 3' end. Same thing with DNA replication that we talked about. You can only add new nucleotides to the 3' end. And by 3' I can kick off because of dehydration. I can kick off this. This is what we mean by we can. There's a phosphate group that's already. So what's going to come next? Now we're talking about mRNA though. U. Good. A. B. B. A. E. U. C. Good. And now we got the 3' end. Okay. So we've now created what we call an mRNA transcript. Notice how similar it is to the coding strand. It's the exact same thing, except instead of T's, we have U's. So this is why we call this one the coding strand, because its literal code is going to end up being the mRNA. The only difference is that instead of T's, we've got U's. That's why we call it the coding strand. Okay. Yeah, so this is the process of transcription. We've got a couple more slides that I'm going to walk through. Okay. So obviously DNA exists as a helix. So before anything can happen, excuse me, RNA polymerase has to unwind it. We can see that happen here. And it's going to be transcribed again in the 5' to 3' direction, meaning new nucleotides are being added to the 3' end. And then RNA polymerase will also rewind the DNA once it is already transcribed that section. Nothing to write there. Okay. So I do want you to write this. This is important. After the mRNA transcript has been completed, so after we've already made this, it has to go through what's called post-transcriptional processing. So after transcription, it has to be processed, and certain things, several things happen to it. Okay. The first thing that happens is a GTP cap gets added to the 5' end of the mRNA transcript. So we would add a GTP right here. And this GTP cap is meant to help the ribosome to recognize the mRNA. We want to be able to, the ribosome needs to be able to recognize it and say, this is what I need to be translating now. We'll talk about that process tomorrow. So the GTP cap's purpose is to help the ribosome recognize the mRNA transcript. And so it gets added to the 5' end. There's also what's called a poly A tail that is added on the 3' end. So keep in mind that once the transcript has been made inside of the nucleus, it's going to have to leave the nucleus and travel through the cytoplasm. As it travels through the cytoplasm, it gets exposed to all these different enzymes that are going to be literally biting off the end. And so we don't want those enzymes to bite off the actual mRNA message. That message is important. We need it to eventually be translated. So what we do is we basically insulate the real message. We're going to put that poly A tail on the end of it. And by poly A, what we mean is it's literally just like potentially hundreds of adenine nucleotides that get added. And if those get bit off, we don't care. That's not a part of the actual message. So as the mRNA is traveling through the cytoplasm to get to the ribosomes, we want to be able to protect the actual message. If we add that poly A tail and those poly A tails get bitten off, that's not a problem to us. They don't actually carry any of the message. Did you get that? Did you write it? ¿Por qué no? Why not? You're giving your hands a rest? Okay. Well, I guess I can make these slides available to you all. Yeah, but we're not going to talk about this. Just take a picture of it. So this is really, I hesitate to use the word ingenious, but this is really a clever process that protects the cell. Protects the transcript, the mRNA transcript. Selena, do you have any questions about this?
No.
Okay. The mRNA transcript has non-coding regions that are called introns. I've always kind of felt like the names of these regions should be switched, but the introns are basically spacers that exist between the exons. Introns don't code for anything, and exons do code for something. This is a big question in science, especially in evolutionary science. Because of the process of evolution, things that don't get used tend to be eliminated over time because the cell doesn't, or an organism wouldn't want to waste its resources or its time making something that it's not going to use. So the idea is that these introns, even though they might not code for anything, the reason that our cells continue to make them probably means that they do still have some function that we just don't yet understand. So this is a big question in science. Why are the introns there if they end up getting taken out anyway? So the introns are basically, from what we understand now, they're basically just spacers. They don't code for anything. And so the cell is going to actually, or not the cell, but in this process of post-transcriptional processing, those introns will get cut out of the transcript in a process that's called splicing. It's a fun word to say. The introns get cut out, and then the exons get bound together. They get spliced together. The introns get removed, and then the exons are spliced together. Once those exons have been spliced together, I'll go back. Once those exons get spliced together, we have what's called a mature RNA. So post-transcriptional processing starts off with an immature mRNA transcript. It's going to add the poly A tail, it's going to add the GTP cap, and it's going to go through splicing. And that creates the mature mRNA transcript. Okay, one more. So we can remove the introns or the exons as well in different variations. And this can result in different transcripts. And different transcripts can code for different proteins. This is a process that's called alternative splicing. We can end up with various combinations of introns and exons that give us different codes. Yeah. Selena, you've got to evacuate the building.
I'm weak.
Okay, I think that was the last slide I had anyway. Let me just see. Hold on, let me just check. Yeah, that was the last one. There is an assignment. It's like an FRQ.
Okay.
I do have some practice questions that we were going to go over at the beginning of class, but I wanted to wait for you, Selena, so we'll just go over them tomorrow. But I do want you guys to please try to do that FRQ tonight, or at some point today. It's just 2 questions.
All right. Did you record the class yesterday?
Yes.
Okay. Where did you put it?
I'll put it on YouTube.
Okay. Like on your channel?
Yeah.
Okay, cool. Because I subscribe to it.
Okay. All right. Have a good day.
You too.
Bye-bye.